The overall goal of this procedure is to measure functional motor impairments in the experimental autoimmune encephalomyelitis, or EAE, mouse model of multiple sclerosis, using kinematic gait analysis. The application of kinematic gait analysis to mouse walking behavior has been previously established and described by others.
Neurological deficits in EAE result from neuroinflammation and sporadic white matter loss throughout the spinal cord and cerebellum. Traditionally, motor disability in EAE mice has been assessed using clinical scoring systems, in which mice are assigned a clinical score based on the observer's impression of the severity of motor deficits. Data from clinical scores are ordinal and do not correlate well with spinal cord histopathology. Kinematic gait analysis has recently been shown to be a better behavioral correlate of white matter loss than clinical scores, in addition to providing an objective description of walking deficits in EAE mice. This technique involves placing reflective markers on the hind limbs of mice and allowing them to walk on a treadmill while being recorded with a high-speed camera. Kinematic parameters are then extracted from the video using motion analysis software.
The first step is to make the markers that will be placed on the animal's hind limb. The reflection of light off of these markers allows the coordinates of anatomical points on the leg to be extracted from the videos. To begin, punch the desired number of circles from the sheet of reflective paper. Each mouse requires 5 markers per recording, 2 large and 3 small. Using fine scissors, make a cut extending from the perimeter to the center of the circle. Peel off the paper backing to reveal the adhesive surface using fine forceps.